Hi, Nutrition 101. Welcome to part 2 of carbohydrates. Today we're going to start digesting a bagel, cream cheese, and some jam based on their carbohydrate types. So we're going to start this with carbohydrates, and then in the next two chapters we will digest the protein and the fat that we also find in a bagel with cream cheese and jam. So first off, let's review. Let's think about the types of carbohydrates. Last time we talked about monosaccharides, disaccharides, and polysaccharides. Our monosaccharides include glucose, fructose, and galactose. Our disaccharides include maltose, lactose, and sucrose. And our polysaccharides include starch, fiber, and glycogen. Glycogen we can just get rid of in terms of starting products for digestion because we don't eat glycogen. It's our storage form of carbs in our liver and in our muscle. And by the time we eat animal, there's no glycogen left in their muscles. So if we're going to consume a whole grain bagel with grape jelly and cream cheese, or strawberry jelly and cream cheese, one or the other, what type of carbs do we consume? So let's think about it. A whole grain bagel made with 100% whole wheat flour. Let's pretend that this bagel has no added sugar in it. And so in this case, we would say that that whole grain bagel was made of starch, would be the first little blank there, and fiber would be the second blank. Then, if we think about carbohydrate in our strawberry jam, we could think about the strawberries, which would contain fructose, and then the table sugar, which is sucrose. And then if we look on the bottom there, our cream cheese is, of course, going to naturally contain lactose because it's a dairy. Let's take a look at the chemical digestion. The starch starts to be digested in the mouth. The salivary amylase is the digester. In the esophagus, the salivary amylase continues to work, but nothing new happens. In the stomach, the salivary amylase is deactivated because it's a protein enzyme. And since it's a protein, it gets denatured by the acidity of the hydrochloric acid in the stomach. In the small intestines, pancreatic amylase is secreted by the pancreas to digest starch. And then maltase, sucrase, and lactase are our disaccharidases in the small intestines, and they continue digesting maltose, sucrose, and lactose. In the large intestines, any thing that hasn't been absorbed at that point gets broken down into smaller pieces, water gets reabsorbed, and excretion becomes a little bit easier that way. Now, when we look at what is happening in terms of this chemical digestion, the purpose of each of these enzymes, our amylase, our maltase, our sucrase, and our lactase, the purpose of those enzymes is to activate hydrolysis reactions. Hydrolysis reactions involve adding water to a molecule to break it apart. So a hydrogen is added to one side of the molecule and an oxygen and hydrogen are added to the other side of a molecule to break apart that molecule. And so our purpose of digestion is to take any of the disaccharides and the polysaccharides and break them down into glucose, fructose, and galactose. We also have a little bit of help with mechanical digestion here. So our mouth includes our teeth, our tongue, and our saliva. Our esophagus has peristalsis, pushing that food down to the stomach, where the gastric juice and the musculature of the stomach slosh and churn and moisten the food. Then in the small intestines, our segmentation pushes food along a little bit more. And in the large intestines, water reabsorption occurs. So when we're in the small intestines, our end products include fructose, glucose, and galactose, our three monosaccharides. These are then absorbed in the villi of the small intestines into the mucosal cells, and then they travel to a venule, to the portal vein, and then to the liver. And once we're in the liver, fructose is more readily used to be converted to fat and made into glycogen. Glucose causes blood sugar to rise, and galactose is converted into glucose through a chemical reaction. So next, let's talk about metabolism. We're going to first talk about when things go right, and then in our next lecture, we'll talk about when things go wrong and give some applications. So let's look at when things go right. We have two types of metabolism, remember. We have catabolism, which is breaking things down into energy, and anabolism, which is building things up. We have a number of vocabulary words that we're going to need to know in order to understand glucose transport and metabolism. Those include the glycemic index, which is how fast your blood sugar rises after eating a certain carbohydrate food, the glycemic load, which takes into account the portion size of that food consumed, anabolic pathways, which involve building structures. In carbohydrates' case, that could be fat or that could be glycogen. Catabolic pathways, which include anaerobic glycolysis and cellular respiration. Cellular respiration is with oxygen, anaerobic glycolysis is without. Insulin and glucagon, which are two hormones that are secreted by the pancreas. Insulin's job is to lower blood glucose when it's high, and glucagon's job is to raise blood glucose when it's low. Lactic acid is the byproduct of anaerobic glycolysis, which is why we get so sore and have to stop after starting out a little bit too fast in a run or a sprint or something like that. So let's talk a little bit about the glycemic index. As I mentioned, the glycemic index is how fast your blood sugar raises after a meal. This little chart right over on the right here shows how simple sugar would cause blood sugar to rise. So just about an hour after consuming a meal, blood glucose levels would go way up. If you're eating more of a mixed meal, that little rise would be the same surface area underneath, but a little bit slower. So your max blood glucose might happen 2 to 4 hours after consuming food. The increase in blood glucose stimulates the pancreas to secrete insulin. Insulin's a hormone. You can think of it as like a key that unlocks the doors to certain cells. Insulin can unlock the doors to muscle cells, to liver cells, or to fat-storing cells, as well as other energy-needing cells there, so like brain cells and nervous system cells, those sorts of things. Once all of the blood glucose is stored in cells, several hours after a meal, the blood glucose level drops, even a little bit before normal. Blood glucose is typically right around 90 milligrams per deciliter. And so when we look at that dip, that might dip it down to 85 milligrams per deciliter or so. That little dip stimulates the pancreas to secrete glucagon, another hormone, which then causes the liver to either synthesize glucose or to break down glycogen into glucose and release it into the blood to normalize that blood glucose again. And so after you're fasted for, say, 12 hours or even 8 hours sometimes, your blood glucose level would be at fasting level, which is actually a little bit higher than those few hours after a meal. So cellular respiration. So as we digest, right, we take that starch that we consume, we break it down to maltose, we break that down to glucose. If we transport that glucose around to glucose-needing cells and add oxygen to it, we can create carbon, water, and energy, or carbon dioxide and energy. That energy is ATP, and that's how we provide energy for the work that our cells need to do. So the more work our muscle cells are doing, the more ATP we're going to need each minute. So our higher work output. So if you're running at an 8-minute mile versus just standing around or just sleeping, then you're going to require more power each minute to fuel that to happen. So carbohydrate metabolism. Anaerobic glycolysis happens in the cytosol of the cell. It's without oxygen. And it has a byproduct of lactic acid fermentation, so we can't use it for very long. You can create about 2 ATP per glucose using that process. A much better, more efficient way, as long as there's oxygen available, occurs in the mitochondria. That's called cellular respiration. The byproduct of that is carbon dioxide, which then we can process and breathe right out, and we create 32 to 36 ATP molecules per glucose molecule. So it's a lot more efficient process. And if we look at this cellular respiration, you can see this anaerobic metabolism happening outside the mitochondria and the cytosol of the cell. And then you can see the aerobic process happening inside the mitochondria of the cell. So oxygen is exchanged with the mitochondria, and that allows pyruvate to be converted into acetyl-CoA, which then causes this cascade of events to happen, including the citric acid cycle. You might also be familiar with it being called the Krebs cycle, and the electron transport chain. both of which produce this highly energized ATP. So that's considered our catabolism. That's taking the nutrient and breaking it into energy. Let's just review where else glucose can end up. So glucose, as you see, that catabolism was an example of the one on the left, the brain and the other cells for energy, and the one on the right, the muscle for energy. Glucose can also be stored in the liver as glycogen. We have about 400 calories worth of storage in the liver. Or in the muscle, we have about 1,600 calories worth of storage in the muscle of glycogen. Glucose can also be converted to fat through lipogenesis, so the glucose molecules are transformed into fatty acids and built into triglycerides, which is how we store our fat in our body. So if you consume too much of any energy-containing nutrient, we have this unlimited capacity to store adipose tissue. So any excess glucose is going to end up as adipose tissue, which is why when you consume too much of any nutrient, it can end up making you gain weight and especially gain fat. So next time we're going to talk a little bit about what happens when things go wrong. So what happens in diabetes, what happens in ketosis, what happens in hypoglycemia. So stay tuned for our next lecture to talk a little bit about what happens when our body's not functioning correctly, we might be missing something, and glucose metabolism can't happen as it should.